Okay, so hello everybody. So welcome to the event Tech Co-Leader, a knowledge share event in collaboration with several partners and also MISE. And I would welcome everybody. I would just wait a couple of minutes more to make all the audience to join the event. We will start the question. My suggestion is for everybody to switch off the microphone until I will invite you to open the microphone, so we will not hear any noise, and we will leave speakers to be free of sharing their projects. So let's wait 2 minutes. Just we will start in 2 minutes more, 2:05, and to let all the people to join. Great, 2:05 exactly. So we can start the second session of the day, and welcome everybody to the session which title is Treatments and Therapeutics When Patents Meet Industry. Actually, it's really a pleasure for me to be the chairman of this event. Basically because my passion has been always to develop drugs, and it's very important for me to be here today. So today we are going to talk about some projects that actually have been submitted to some grants application. They made some grant applications from MISE and then have been awarded. And that's it's a proof of concept grants, which is really important. So today I'm going to introduce briefly myself, what is the topic of the session, and then I will give the table and everything to the speakers of today's. They have the time to introduce themselves and to introduce the project. At the end of the project, after the presenters, they will present their project, there will be a panel session where everybody, also the people online, they are allowed to make questions. And actually, we are really happy to answer to the question that you can make directly to the speaker and to myself as well. So who is myself? So my name is Giovanni Rizzo. I'm today the partner and key manager of biotech fund in Indaco Venture Partners. I've been studying and working always in the field of drug development. My background, I'm a biologist. I have a PhD in molecular oncology. I did a specialization in microbiology. I did a master in drug development, and then I did an MBA in London. Work-wise, I have been a researcher for many years. Working on leukemia first, and then I moved in gastrointestinal and liver diseases, where I joined a freshly made startup called the Intercept Pharmaceutical in between Perugia and USA, New York. I've been the head of biology since the inception of Intercept Pharmaceutical, so from 2002 till 2014 almost. And then I moved to London. I was working in another venture capital for 1 year called Medici as a vice president of a startup in Cambridge. And then I moved to the UCL, where I had an honorary senior lecturer position for 7 years, and there I joined a startup called The Accurate, where I was Vice President Research and Development, and we made a deal with Takeda. And then I came back to Italy. I was the head of innovation of Zeta Cube, which is a branch of Zambon, and in that activity, I was actually building an accelerator of startup, open accelerator, an international accelerator for startup in the health tech. And then I moved from Zambon. I became the CEO of a portfolio company of Indaco Venture Partners, and we made a deal with a company from California. And then we started doing fundraising for Indaco biotech fund. And actually, I'm now today partner and key man of the fund. A few words on Indaco. Indaco is the largest VC in Italy with 5-6 funds under management, investing in broad e-commerce and also digital medtech. And recently we launched the biotech fund. So today we launched the fund in June 2022. So we closed the first round about it's 97 million. We are still in fundraising. Indaco Biotech Fund invests only in therapeutics, investing in early stage and Series A. It's that they go from 200,000 up to 5 million. And the allocation of the capital is 75% allocated to Italian companies. 25% will be allocated outside Italy. Other than me, the Indaco Bio, it's constituted by 3 other partners: Davide Turco, which is the CEO and managing director of the managing company Indaco Venture Partners; Elisabetta Robinson, which is the vice chairman of Indaco Venture Partners; and there is a third partner, which is Gonzalo Andrade. It's a Portuguese person that I met a long time ago, always in the field of, he worked always in the field of life science. He also built a small VC fund in Portugal. Adding to those people, there are 3 other members of the team, investment team, which is Elena Beltrami, that is an ex-Teleton and PhD with the MBA as well. Vittoria Pavoncello, that comes with an experience, is another VC called Science, and she's also in as a background, technical background, with the PhD, the master in neuroscience. Lucas Seal, that it's an associate, and actually it's has a business background, but has been involved for a long time with a pharmaceutical company in the distribution, so he knows very well also the business behind the project in the life science. So, and that's what is about myself. So regarding the topic today, it's very important for me to be here today, mainly because the scene, the Italian scene of the biotech, actually, it's changing quite fast and quite quickly. So you have seen recently that in contrary of what is happening in the rest of the world and in Europe, actually Italy is pushing a lot in the field of the life science and biotech. There have been many funds that have been created recently. Other than Indaco, there is Oxygen, there is Panacas, Claris, Aurora. So there are many funds in the field that actually can invest in the biotech. And this is very important, thanks also to the support of the government with the CDP that is actually allocating a lot of capital in the funds. This is increasing the chances that the university and the entrepreneurs like you can actually get funds. And my suggestion is not to see anymore at the thing that actually is very difficult to find money in Italy. It's not difficult to find the money. There are money that can support entrepreneurs. Now we need to find the right way to unlock those money, and this is very important. So those kind of events are also important to share the things that things are moving in Italy, are moving fast, and we need to create that ecosystem which is healthy, but at the same time can push for the innovation to start and to be at least close to the one in Europe that are investing a lot and are actually proceeding proceeding very well, like UK, Germany, Netherlands, and France. So that's overall. One last comment that I want to say on the title, and then I leave the stage to the presentation. It's the title is quite interesting. When patents meet industry, there are two ways to understand what is the title about. One is, when is the time to make the patent? So we speak a lot about the patents, but the patents is something extremely important. And I strongly, and this is my personal point of view, I strongly believe that the patents, they need to be done, they need to be filed when actually you have the chance and you have the wish to make a product. If you make a patent and you don't pursue the way to make a product, I think that can be also detrimental for overall the system in the world, because somebody else can make a new patent and actually can develop a program and project. So my suggestion is, yes, make the patent, but it's not a starting. It's not the end of the story, but it will be a starting point for you to build something and to make a project and a product. And the second point is that patents are meeting industry. That's true. Patents can meet pharmaceutical in this case, but patents can meet also venture capitalists or other people like business angels or incubator accelerator. And actually, the point is that just look 360 degrees where you can get the best out of making the patent to become a product that can be a business angel, VC, incubator accelerator, and the pharmaceutical company. Altogether, we try to actually to work to make this ecosystem working properly to make this to happen. So I close my presentation here, and I want to give the stage to the first speaker of the day, which is Professor and medical doctor Italo Linfante. Just introducing a bit himself, and Italo, it's actually medical director of the interventional neuro radiology at Miami Neuroscience Institute, and it's also clinical professor of radiology and neuroscience at the Herbert Wertheim College of Medicine in Florida. So I will leave him the stage, and thank you, everybody.
Hi, good morning, everybody. Good morning, Dr. Rizzo. Thank you very much for having me. I don't know if you can see my screen. Can you guys see my screen?
Not yet.
I'm going to give you okay, you are presenter already, so you should, okay, perfect now, yeah.
See the screen?
Yeah.
Perfect. Very nice. All right, so thank you for having me. I'm gonna show basically two parts. The mostly our field of interventional radiology and vascular surgery is driven by technology and by development. I've been doing this for a while, so besides taking care of patients, doing operations, I work with several startup or big companies to develop new products. And I'll show you the journey over the last 20 years. Be very exciting in this field. So these are my disclosures. As I mentioned before, I work with several companies in different areas. So I work at Baptist Hospital, which is Baptist Health South Florida, one of the largest healthcare organizations in South Florida and in Miami. And in particular, Miami Cardiac and Vascular Institute is one of the largest vascular institutes. So what is it that we do? This is our case volume of operation that's been growing up tremendously. We are doing almost 100 in January, we do almost 100 cases operations a year. So it's a field which is growing tremendously thanks to innovation and technology to give our patients much better outcomes. So I'm going to show you, sorry, quickly the journey from imagination to reality, like you know very well, and Dr. Rizzo presented very well. So what is the journey from an idea to realization? I'm going to talk about an old story and a new story. So the old story started with a patient with a 56-year-old woman who developed the worst headache of her life, followed by violent nausea and vomiting, quickly unresponsive. She has a horrible exam, terrible neurological status. And the reason of this is because she has subarachnoid hemorrhage. So this is a CAT scan. You can see the eyes of the patient, and the white things here not supposed to be here. This is blood. So she bled inside her brain. What is she bled inside her brain? Because an aneurysm ruptured. The incidence of subarachnoid hemorrhage is ranges from 2 to 16 per 100,000. Old age adjusted incidence rate in low to middle income countries was almost double that high income countries. United States is about 9.7 per 100,000, but it's a deadly disease. So median mortality in the United States was about 32%. It's up to 55% in Latin America, 43% in Europe. So it's a bad disease. But the good news that mortality rates from aneurysmal subarachnoid hemorrhage have declined thanks to the work of and the collaboration between industry and us physicians over the past 25 years. I'd like to show this slide from my colleague Andy Molyneux from Cambridge that shows rates for million population in terms of mortality. And you can see that at a certain point in 2002, that's why I told you it's an old story, everything dropped dramatically. So the mortality for aneurysmal subarachnoid hemorrhage is going down tremendously. And why is that? So what happened in 2002? So in 2002, there was an approval for a revolutionary treatment in securing the ruptured aneurysm. The traditional treatment is traditional surgery that involves a craniotomy, and the surgeon does a dissection of the sylvian fissure, find the aneurysm, and then put a clips on the aneurysm. Of course, it's very invasive. So imagination to reality was that how can we do this without doing a craniotomy, without opening the skull? And we came up with endovascular embolization. So basically, we access the femoral artery of the groin, and we thread a little catheter all the way up into the brain. And then we insert inside the aneurysm, which is like a small pouch, is an outpouching of the artery. It's a weak spot in the artery. We introduce detachable coils initially. That's how we did initially. And the detachable coil closes the aneurysm. So basically, we secure the aneurysm without opening the skull. And of course, this works beautifully. So it's one of the cases that I showed you before. Let's say an artery of the brain, tirocerebral artery. That's the aneurysm. It looks very ugly looking. And we're able to place coils inside and close it and secure it. So with this approach, they really revolutionized the treatment of cerebral aneurysm. There's tremendous amount of data now of many hundreds of publications. So the endovascular approach to treat brain aneurysm is more safe and effective compared to traditional open surgery. And then, of course, once you open a road, as Dr. Rizzo knows very well, then you can develop better technology to make this procedure even more safe and effective. We develop stents, we develop better coils. There was a question of recurrence if the aneurysm stays occluded or tends to come back. There was a question what we're going to do for large and giant aneurysms, because not all aneurysms are what I showed you, and about dissecting aneurysm and blister aneurysm. We're going to talk about that. So this is a giant dissecting aneurysm. Is a young woman. It's a 16-year-old woman. Developed general tonic-clonic seizure, headache, and nerve palsy. On CT, the aneurysm was so big that they thought it was a brain tumor initially. But then she had a giant internal carotid artery aneurysm. So as you can see, that's the skull of the patient. The artery comes in, internal carotid artery, and it's all completely blown out by this dissection, giant dissecting aneurysm. That's how it looks like. That's a video I want to show you if it starts. We can see that the contrast, it's blood that goes inside this gigantic bomb inside a 16-year-old girl. So this was absolutely untreatable. There's no way you can do surgery on something like this unless you want to close the artery and give the patient a massive stroke. And coils don't work. So we developed a different device. We've actually working on this before on a different device. Like I say, imagination to reality, and we came up with the flow diverter. I had the opportunity. There was me many years ago, working with Dr. Wakalu up in Boston, and we had this idea of creating a stent, a very tight mesh stent that could divert flow from the apparent artery into the aneurysm, and therefore securing the aneurysm. That's how the prototype looks like. Very flexible stent. So a standard diverse flow from the aneurysmal sac into the parent vessel will change the fluid momentum transfer into the aneurysmal sac and produce endothelial growth. This is the, so in biotech, we're going to talk a little bit biotech at the beginning, and then we're going to talk about drug development. In biotech, we start with engineering. It's like, building bridges or something. So it comes with mathematic idea first, and then you develop a prototype, and then you move to the animal model. This is an animal model of aneurysm. You can see the device placed inside this artificially made aneurysm. There's a clot formation. This is histology. And then after the clot gets organized, then the endothelium grows over the device, securing therefore the aneurysm. We published in 2007. But then several papers and hundreds of patients treated with this technology back 2009, 2015 show the technology is safe and effective. So what about our patient? In our patients, what I did, I placed multiple of these devices, reconstructing the artery, from the middle silver artery all the way into the telecarotid artery. And you can see the mathematical modeling theorizing that after you place the device, there's contrast stagnation inside the aneurysm, means that the blood stagnates inside the aneurysm and forms a clot and secures the aneurysm. That's how it was before, and that was after. You can see the artery is completely reconstructed with a new scaffold. We basically reconstructed the artery from the inside. That's what this technology does. Let's see aneurysm before, aneurysm after, aneurysm before, aneurysm after. So what happened to the patient? 5 years later, beautiful young woman. She was modeling for the American Heart Association, Go Red for Women, telling her story and fundraising for improving our technology and doing fantastic. Strong of this experience, we published recently a very large case series of endovascular treatment for giant endothelial aneurysm. And I remind you, those were untreatable and still are difficult to treat unless you have this technology and are expert in doing this technology. And the paper was published. So another patient, 15-year-old man, was the leg of his life, general circulatory seizure, loss of consciousness, subarachnoid and intraparenchymal hemorrhage, so a ruptured aneurysm. Another bomb inside the brain, the completely horrible looking dissecting middle subarterial aneurysm. The artery enters here, gets completely blown out and comes out at a certain point. That's how it looks on angiography. Reconstructed with two flow diverters. And after reconstruct the position of the flow diverter, you can see the stagnation inside the aneurysm and the beautiful visualization of both branches of the middle subarterial. Aneurysm before, aneurysm after. 6 months follow-up angiography, patient did extremely well. Complete reconstruction of the artery and exclusion of the aneurysm. For blister aneurysm, we published on that too. We were the first ones using this device for blisters. For a blisters, they're very rare aneurysm, particularly very, very small aneurysm. They're rare but very deadly. I don't want to go too much on it. Cannot clip those because the artery is completely disrupted. There was one of the first patients that we did many years ago. It was a young woman. She was actually a physician. Developed this subarachnoid hemorrhage. So we try to worry about small rupture aneurysm. So in this case, in angiography, you can see the teeny tiny microscopic aneurysms on the surface of the middle silver artery of this young woman, which is of course impossible to treat with any other technology besides flow diversion. So we tried flow diversion for the first time. There was a flow diverter position in this very small vessel. These are like 1.5 millimeter vessels, and the results was beautiful. Just after the positioning of the device, there's a poor visualization of the blisters and good reconstruction of the artery. Patient did fantastic. 8 months follow-up angiography, complete reconstruction of the artery, as you can see here with the device inside, and occlusion of the aneurysms. How did the patient do? She finished residency, fellowship with oncology, married, 2 children. And then, strong of this experience, published 2 papers, one on a small case series just from us, and then we did a very large series with other investigators. And now the treatment for both blister aneurysms is basically flow diverter. So we demonstrated from idea, imagination to reality, from the mathematical model, a prototype, to clinical trial, and clinical experience showing that this technology works. So, in summary, flow diversion is an excellent option in large and dissecting and cerebral aneurysm. Surface coated technology is advancing, and we're working on surface coated flow diverter to prevent clotting, and smaller flow diverter for small arteries. So this is the new frontier, surface coating this device. I won't spend too much time on that, but that's the new frontier of this technology. What about the news story? The news story, it's about ischemic stroke. Ischemic stroke is much more common compared to subarachnoid hemorrhage from many reasons. It's basically so this is the cerebral angiogram of a patient. You can see that the artery present on the right side, but there's no arteries on the left side. So basically there's a clot that blocks the flow of blood in one of the vessels. They're estimated to be probably 500,000 acute strokes per year in the United States, maybe 200,000 in Europe, or close to 1 million in China. So it's a very common disease, and disease was terrible because once you block access of blood to the brain is severely damaged. You can see in this case. We have some damage, some assessment of the damage caused by stroke. Patient comes in, and we use this NA stroke scale, which is a neurological examination in points from 0 to 30. So if a patient has an age of 17, the risk of death or disability is 84%. Occlusion of the main artery of the brain, called the middle cerebral artery, results in 80% mortality. Occlusion of the carotid, 73% mortality, and so on. So how you resolve this problem? With revascularization. Of course, the artery is blocked, you have to open up a basic plumbing. We published that it was an independent predictor of good outcome in multivariate analysis. And so we had to come up with a way to open the artery. So using the same endovascular approach, we reached the artery which is blocked, and it was the idea, the imagination, to create a device almost like a stent that grabs the clot, and you flow arrest with a balloon guide cutter, and you extract the clot and recanalize the artery. Does it work? Yes. In 2015, we published. There were 5 clinical trials published in New England Journal of Medicine in several parts of the world. In the United States, we participated in the SWIFT Prime. It was a United States trial that shows that that endovascular thrombectomy. That's how this new procedure is called. We come up with a new procedure basically. Is able to recanalize the arteries quickly. A 95-year-old, atrial fibrillation, left hemiplegia, neglect, and deviation, an age of 18, occlusion of the middle cerebral artery, very tortuous vessels. You can see the artery is blocked. There's no arteries. That's the blockage. And then we're able to, with this technology, to navigate. There's a catheter you can see navigate into the blocked artery. You can see it looks like it's floating in the air, but it's actually inside the artery, which is blocked, but you don't see it because there's contrast stagnation. We're able to position these devices all the way up into the brain vessel pretty quickly, actually. This is in real time. We can do this procedure in about 20 to 25 minutes. Most of us who are experienced operators. And recanalize the artery. So you can also overcome difficult tortuous vessels. So that's the artery recanalized. You can see these beautiful vessels afterwards. And this is imagination to reality. There was a design initially. That's actually exactly how it looks like. It is my hands holding the device, a stent retriever with the blood clots attached to it. So it does work. How this patient did very, very well. 28 minutes, punch or two recanalization to open the artery to recover. Now the patient, 72-year-old man, atrial fibrillation, hemiplegia, aphasia, and age of 17, so pretty bad, T occlusion, carotid occlusion, intubated in the angio room. At this point, you see the artery is blocked in the neck, but this used to be a no-no when I started this job. Nobody would touch, no vascular surgery would touch a closed carotid, rightfully so, but we can open those. Again, with these devices, we're able to navigate, you can see in real time, past the carotid occlusion, navigate, reach the middle cerebral artery, and then advance the device. As you can see, this is us advance the device from the outside into the patient brain and reach the middle cerebral artery. That's the artery after the recanalization, beautiful results, and the carotid in the neck, beautifully recanalized. That's a clot. So how did this patient do? A 72-year-old from 18, AD to puncture 45 minutes, puncture to recanalization 35 minutes for this procedure, and patient did very well. Okay, so we are in a revolution in acute stroke treatment, and that's one of introduce you the newest idea, and that's about drug technology. I mean, drug development, medication development. So we went in large vessel occlusion for stroke. We went from 80 to 90% depth of disability to maybe 10, 50% and 20 to 40% minor disability. But the question is that we are facing now: how come we open the artery 95% of the cases? It's very unlikely that we cannot recanalize an artery, but we don't have 90% good outcomes. And why is that? We published on that. What are the predictors of mortality? And some patients actually still die from a stroke despite successful recanalization. We published in the journal Stroke. And the reason why this is happening, because once you block an artery, there's an area of the brain which suffers. It's called the core of ischemia, and an area of the brain which is underperfused, which is kind of hanging in there. So once you recanalize, you stop the progression of core. But if you don't recanalize the artery, then the core expands. And there was a beautiful data from JAMA, Journal of American Medical Association. They showed that there's a tremendous decrease in good outcomes as time goes by, because as time goes by from time to onset, symptom onset to recanalization, the brain suffers and these patients don't do very well. So despite recanalization. So we figured out that in acute stroke, brain dies very fast. The previous estimate was 1.9 million neurons per minute. But figured out there's high variability. So some patients are slow progress, so they progress very slowly, about 35,000 neurons per minute death. And some patients are fast progressive, like up to 27 million neurons per minute, all published in Stroke. So how can we protect the penumbra and slow down core expansion? I need to be fast, of course, with everything, but the new field to what else we can do to buy time is neuroprotection, and then we talk about medication development. So possible target neuroprotection, there's several targets in the ischemia-induced cellular neurotoxicity that gets activated from ischemia. Another one that we have been working on with other colleagues is augmenting collateral circulation, because when artery is blocked, another vessel comes over. It's called collateral circulation, which is very effective in some patients, especially when they're young, but it's not very effective in other patients. So how can we turn every patient into this patient with a beautiful collateral circulation? How can we augment collateral circulation? Or how can we do both? And so I've been involved in developing with industry pleiotropic agents. So agents, pharmacological agents that act on both. Increase blood flow in collateral circulation, protect the penumbra, and also protect the cell, the neurons. We published a nice review on this subject that I wrote in 2021. You can actually, if you're interested, you can get all this literature. So let me go back to basic science of clinical application. When I first started my journey in the United States, I was working at the NIH. I was working on rats. That was my job. And so they were in rats, there's a middle subartery occlusion model. So you go from the external carotid artery to internal carotid artery, and you close the middle subartery. And that's at the rats. This is a rat brain. Just like us, they have a core and they have a penumbra. And the core is mostly in the basal ganglia, is a rat brain, and penumbra is in the cortex. And turns out the rats have collateral circulation as well. And this is a beautiful work of my colleague, Dr. Cipolla from University of Vermont. It's a rat brain. Rats have smooth brains, not like ours, but a collateral circulation. So she can harvest those tiny microscopic vessels, put them in a petri dish and analyze them, and find out which is the best way to dilate the vessel, and find out the physiology of, or the pathophysiology rather, of the collateral circulation at the microscopic cellular level. So we come up with this compound, carboxyhemoglobin oxygen carrier, also called PP007. So what is this? So as you all know, physicians know, the oxygen is carried in the bloodstream by the molecules of hemoglobin, but is inside the red blood cells, which is a big cell. So we can extract the hemoglobin, conjugate it with a little polymer by a process called pegylation. So at this point, the hemoglobin molecule is not inside the red blood cells. I mean, of course it is, but in this compound is outside the red blood cells and conjugated with this polymer. And then we add carbon monoxide. So basically we turn the hemoglobin into oxygen transporter, which is exactly what hemoglobin does. And that's how this compound looks like. looks like. So of course, because the hemoglobin is not in this compound is not contained in red blood cells, it's very easy to transfer from the artery into the tissue, because it doesn't have to cross the membrane of the red blood cells and other reasons. So it can go into tiny microscopic vessels and then diffuse much more efficiently compared to a red blood cell. So there's three mechanisms of action of this compound: deliver oxygen more efficiently than a red blood cell. Carbon monoxide induces vasodilatation, so dilates the artery. The problem when you ever dilate the artery, there's hypotension, but this doesn't happen because the pegylation confers a plasma expander property. So actually we have an increase in blood pressure when we give this compound. So theory is beautiful, tested in animals, beautiful work by Ray Kohler at Johns Hopkins University. He measured peel arterial diameter, so measuring actually the changes in the how the electroangiostomosis, the collateral circulation increases in volume once you get this compound. We test several compounds, of course, and the compounds that comes on top. We see the little triangle here, pegylated carboxyhemoglobin. As you can see, the vessels go down during condition of ischemia, but when you give this compound, they stay up. So they're dilated. And this, of course, corresponds to a very low infarct volume. Of course, if you protect the penumbra, once you recanalize the vessel in this type of rats, you end up with a smaller infarct volume. And then we published with Marilyn Cipolla in JCBF in an interesting model in chronic spontaneous hypertensive rats. So these are rats genetically modified to develop hypertension at 4 months of life, very short lifespan, of course. They're very poor electroangiostomosis, poor collaterals, and as soon as you close the artery, they progress very fast. So this is kind of the correspondence of fast progressors in humans. And nothing works in these rats. So we did experiment. There's a rat's brain, use laser fluorimetry probes to measure blood flow in the collateral circulation. Filament goes in, blood flow drops, of course. And then when you open up the artery, blood flow does not recover in placebo-treated rats, but it recovers beautifully in rats treated with this compound. So the ability to increase collateral flow with PP07 might be useful at adjunct to endovascular therapy, and therefore I designed actually on the principal investigator of the United States, a trial. So you go from the model, the idea, to the basic science, experimental ischemia in animals, to convince the FDA to grant us a trial. So the trial is a phase 1, safety. Study objective of EMERA-1 was safety and tolerability of PP07 in acute ischemic patients. And we have secondary outcomes, like every clinical trial, pharmacokinetics, effect on collateral blood flow, etc., etc. The trial was designed as a randomized 3-to-1. So the patient meet the eligibility criteria. Patient got the infusion of PP07 plus thrombectomy, and the controls got just thrombectomy. And then we do all these measurements. And we had 6 investigational sites in the United States, 16 subjects, 3-to-1 randomization, etc., etc. Spare the details. Clinical sites, they collaborated with us, Baptist Health Miami, University of Pittsburgh Medical Center in Pittsburgh, Grady Memorial Hospital in Emory University in Atlanta, Hospital in Jacksonville, etc., etc. We assessed safety as mandated by the FDA with an organism called Data Safety Monitoring Board, DSMB, which is of course completely independent from us. So the DSMB in the United States, that's how clinical trials are done, evaluates outcomes. They reviewed all the data over 3 meetings, and they didn't see any safety issues with the medication given to patients undergoing thrombectomy acute ischemic stroke. Pharmacokinetic studies showed measurable level of this medication sustained over 24 hours, which is amazing because this compound hangs around for 24 hours, which is remarkable. They deliver oxygen in area of ischemia up to more than 24 hours. And we found that the anti-stroke scale at day 7 was significantly lower in patient treated with this medication compared to patient treated with placebo. These are the data I'm going to show very fast. At 7 days, patient treated with PP07 have better outcomes compared to patient treated with placebo. But of course, the study was not designed for efficacy, it was designed for safety. So this is a kind of a secondary outcome, but it's kind of interesting. Pharmacokinetics of this compound stays at very high plasma levels up to 24 hours. Okay, in conclusion, I'm done. Collateral augmentation and target oxygen delivery are promising strategies to improve outcomes in the aeromechanical thrombectomy for acute stroke patients. Pleiotropic agents, so agents that act on both collateral circulation and cells, seem to be beneficial. We are going to a study extension to include patients with thrombolytics, and then we're going to go for a big efficacy trial, 2B3 trial. I really want to thank my collaborators, Ray Kohler from Johns Hopkins, Chen Kayada from Harvard MGH, Marian Cipolla, University of Vermont, and the steering committee of Raul Nogueira from UPMC and the Libeskin for UCLA. This is us in the angio room, opening a patient and giving the medication. Show quickly the data, 91-year-old, one patient, and it showed 22, aspect of 9, so very bad stroke. You can see occlusion of the middle cerebral artery. Large penumbra area, so we don't do anything that would have been a stroke this patient would have done. Enroll and randomize, reanalyze at 14-12, and then we give the medication. That's the artery before. I did the case myself to make sure that the procedure was effective, and then we give them the PP007 hanging here in the angio room, given to the patient while we're doing the procedure. What did the patient do? 91-year-old, NH of 22, so there's no chance for this patient to do well. NH of 2 the day after, NH of 0, normal exam at discharge. So this is what the stroke was supposed to be like on this patient, and that MRI is basically almost normal. So I'm concluding, imagination to reality and vascular embolization of brain aneurysms was a crazy idea, but it's now it's reality. As if I was mechanical thrombectomy for ischemic stroke, and the neuroprotection perhaps, hopefully, a new promise for acute stroke patients. And I'm done. Thank you very much. Sorry, I was a little bit too long.
Okay, thank you very much, Dr. Linfante. It's very, very exciting, a lot of work. I just want to say that I have questions, but we are going to make at the end of the session during the panel. So please stay online, and that I'm really curious to hear something more. So I would now give the stage to the next speaker, which is Professor Alberto Danielli, which is from the University of Bologna. He's going to speak about a project called the Phase KISS, and it's one actually of the winner of the POC from Mise. Please go ahead, Alberto.
Yes, if you can,
yes, I will.
Please share the. Yeah.
I will. Yeah, I will now. Yeah. Okay, you are. No.
Okay.
Yes.
Thank you very much. So thank you, Professor. I hope you can see the screen. Yeah, and thank you, Professor Linfante. A very inspiring talk, and thank you for the organizer for giving me the opportunity to showcase our technology, which is indeed about phages, and it's a modular vector platform for sonodynamic therapies and applications. Sorry again. Myself, I am in the molecular biotechnology lab, so I am a molecular biologist of the University of Bologna, but much of the work that I've been showing today is in collaboration with Professor Cavallesi, which is in the chemistry department next door. So briefly introducing our technology. So it's vector based on bacteriophages. Bacteriophages are viruses that infect bacteria, and particularly this is 13 phage, famous for phage display for those of you who are in this business for the maturation of antibodies, completely harmless to eukaryotic cells. And so we can, through molecular techniques, just engineer the phage in order to target to target cells, and retarget the phage to, for example, bind to cancer cells, provided there are antibodies or peptides that bind specifically to markers, cancer cell markers. But we also can engineer the phage to target other bacteria. Now, this phage has a very large surface on the capsid, which can be chemically functionalized with sensitizers that can then be hit by light, or in our technology by ultrasound, in order to produce a burst of reactive oxygen molecules, which then have a cytotoxic effect. And this cytotoxic effect is local because the vector has been redirected locally to the desired site of action. And of course, sonodynamic therapy has an advantage over photodynamic therapy because the ultrasound can penetrate deeper in the body. So and when he participated to the POC call, we got that, and it was clear to us that we needed, because of the call, to increase our technology readiness level. And so go from 3 as an experimental proof of concept on cancer cells and bacteria, up to 5 in a demonstration and a more validation in a more relevant environment. And for that, we had to really decide and choose better the relevant experimental model. We were aware that we had to go from lab irradiation sources to more clinical irradiation sources, possibly irradiation sources that have a CE mark in order to be used in the European community at least, validate our technology in 3D models and not in cell cultures, and finally try to also move towards the in vivo, at least reach the ex vivo, and demonstrate that when clinical ex vivo sample the technology was valid in order at the end to have it's solid enough to be licensed to a spin-off company. So briefly, we decided to focus the technology with the POC grant on EGF are overexpressing tumors. These tumors, many tumors overexpress EGFR. And so we are actually working on 4 platforms of the vector. One is EGFR targeting, other phages are targeting neuroblastoma and epithelial sarcoma, which are rather rare cancers, but EGFR positive tumors are very frequent. And so this, I will present just the data on this. And so what you can see now, the design of the experiment, we have the phage, it will be engineered, it is engineered to target EGFR receptors. And then we actually excite the sensitizers either by light or by ultrasound. Light as a benchmark, ultrasound as our patent. And as you can see here, we have 2 cell lines. The A431 cell line is overexpressing a lot of EGFR, and we have a negative cell line, which is our benchmark negative control, which does not express. And as you can see from the pictures here, our phage here in pink and purple targets very well the EGFR overexpressing line, but it does not the RH4, and this was also demonstrated with the flow cytometry. So our platform is really specific. Next, we asked, can we kill specifically those cells that overexpress the EGFR receptor? And here you see the experiment with light, and indeed we could see there is a very significant difference in the killing of those cells overexpressing EGFR, and this is also dose dependent, and these data have been backed up with a lab sonicator. Although still we see a dose dependency, the intensity though is and the dying out, the survival is a little bit lower, is worse than with light. So it was clear that we needed to move towards more, clinically or therapeutic devices, or devices that are used in clinic and not stuck with our lead lamps or lab sonicators. And for that, so we adapted the technology to clinical therapeutic lasers, and also using CA marked sonotrodes in order to provide the ultrasound stimulus. And as you can see here, we were able to obtain really good killing of cancer cells, specific killing of cancer cells, using just 1 minute of these sonotrode irradiation. And this is quite striking, because these devices are no fancy high focused ultrasound devices. These are very cheap devices that are licensed for use, even not by doctors, by physicians, but also by when you get a physiotherapeutic treatment in order to amend and cure your muscles after a sport injury, and just 1 minute of it together with phage provides really a straightforward and strong killing of cancer cells. And the phage per se has no whatsoever cytotoxic effect. And next we ask, okay, can we move further towards a more preclinical model? And so we made a cancer spheroid, so small tumoral masses in the lab. And we verified that our vector is able to penetrate quite fastly, and in 2 hours it gets into these spheroids and penetrates deep into the tumor masses. And this is also a quite striking feature in the fact that the CAR T cells do not enter there, and also chemotherapeutics or antibodies do not reach this level of penetration. This is because of the phage biology, but I won't go into that. So now the next question is, what happens if we shine a light or we irradiate with the ultrasound these spheroids that have been treated with our vector? And the result is quite striking. The tumor masses, lab tumor masses spheroids, they just burst. As you can see here, this is the control, perfectly made spheroid. Here we have the burst of the spheroid also with ultrasound, and the burst is accompanied by a significant cell death of the cancer cells. Again, these are 1 minute irradiations. And next we ask, can this technology be useful on more, on patient samples? So this is an ex vivo experiment. These are basically acid fluids which contain natural cancer spheroids from patients affected by ovarian cancer. You see these spheroid little tumoral masses found in the acids of these patients, well formed. After irradiation, these natural spheroids just burst and are killed efficiently by our technology. And what's next? This is still running now. We are moving towards in vivo now with mice. A very talented postdoc was sent of the University of Helsinki in order to be trained on mice. What you can see here is the biodistribution of our phage, so targeting tumors in the mice. So this is ongoing. I have no results to show. But so I hope I have convinced you that thanks to the POC grant by Mise, we were able to reach actually TRL 5, and so our technology matured a lot from the start point, and indeed it became interesting for a spin-off company. So Highly Sense, this is a spin-off company that will be soon starting and at the University of Bologna. So the University of Bologna will be participating to the spin-off company with Behold, which is a major player in the spin-off market or spin-off environment in Italy. This spin-off will be taken. Our patent, the patent will be licensed together with a portfolio of other patents, and this spin-off is dealing with the application of phages of light and of ultrasound for therapeutic, theranostic, and biosensor applications. So I think this is all, and if you're interested in this technology, if you would like to know more about this this spin-off that's going to be starting soon, I think late summer or beginning of fall, please contact me. I'm Alberto Danielli. I am actually will be part of the panel of the scientific committee of this spin-off company, or contact our knowledge transfer office. And this said, I thank you for your attention, and thank you very much for listening.
Thank you, Alberto. It was quite clear, and thank you very much. Question also here. We just waiting for the panel to make the questions. And I would give the stage to the next speaker, which is Professor Stefania Raimondo. So Stefania is assistant professor at the University of Palermo, and it's presenting a solution in collaboration with the Scuola Superiore di Sant'Anna as a drug delivery system for anti-cancer treatments.
So good afternoon. I hope that you can see my screen. Can you please confirm?
Yes.
Okay.
Yes, yes.
Okay, perfect. Thank you for this opportunity. Today I will present you the results of the project that was financed by the POEC. And it is based on one of our past research on the oligonucleotides complementary to a phospholipase, DDHD1, that we have found to be a key, to have a key role in colon cancer progression. So I am Stefania Raimondo, and I am assistant professor at the University of Palermo, and I work in the laboratory coordinated by Riccardo Alessandro. I took my master degree at the University of Palermo, and then my European PhD in Palermo, but also in Sweden. But what really encouraged me to go deep in the technology transfer process and in the patent stories is that in 2016 our team also was the co-founder of a spin-off, an academic spin-off, and for this reason I really work a lot in the technology transfer field, and this is also the reason why today I am an Italian local representative of the EIT Health Innovator Community. So that is a European community of young investigators that want to do innovation in the healthcare systems. So what is the state of art? As I said before, our story started in 2018 when we used to study a phospholipase that was important in colon cancer progression. And from our research, we have patented a technology that was mainly focused on the use of oligonucleotides complementary to the DDHD1 sequence in order to inhibit and induce cell death of cancer cells. But we take advantage of one part of our patent that was the possibility to encapsulate these oligonucleotides into lipid vectors. Why? Which is this challenge today of using oligonucleotides in general in the clinic. This opportunity is hampered by the problem of the systemic administration, because oligonucleotides are unstable, they are poor available to the tissues, and they can have an unspecific targeting. So the need today was to develop targeting systems for bioactive molecules in the organism, including oligonucleotides as well, with the aim of enhancing their biological effect and increasing their stability. So this is the reason why we applied for the POEC, to give us the possibility to develop a technology to deliver these oligonucleotides towards cancer cells. So our solution is based on extracellular vesicles, in particular on extracellular vesicles from plant matrix. They are lipoproteic nanostructures that are physiologically produced by mammalian cells, but also from plant cells. They are a rich complex of biofunctional molecules. And one important opportunity is that they are released by cells, but they can be internalized by target cells, affecting their phenotype. So our aim was to use these nanovesicles from plants in particular in order to load exogenous short interference RNA and oligonucleotides for anti-cancer therapies. In order to do that, we had the opportunity to collaborate also with the spin-off, which aim at producing at large scale these plant extracellular vesicles. And thanks to this collaboration and thanks to the POEC grant, we arrived to a TRL5. So starting from the first results, we have decided to isolate this extracellular vesicle from tangerine. I am from Sicily, so citrus fruits in general are the most available in our land. So we have isolated and characterized these nanovesicles that, as you can see, have a diameter of 150 nanometer, and we also perform some transmission electron microscopy, but also some other characterization assays in order to have a look at these structures and at the morphology of these structures. Then we have worked a lot on this technology. How can we be able to load oligonucleotides into these extracellular vesicles, not attaching these oligonucleotides outside the vesicles, but inside them, in order also to have the possibility to control the amount of the loaded short interference RNA, but also the release. And after this year of studying, we came across the electroporation process. So we have established technology that was mentioned in the patent, but we think that we have improved what we have already presented in the patent, and we established this technology in order to electroporate these vesicles with the short interference RNA, in particular fluorescent short interference RNA that we were able to quantify. And as you can see, we obtain a dose-dependent increase of the fluorescence that was directly dependent on the amount of short interference RNA that we load inside the vesicles. Then what we did was to incubate this complex, the complex of extracellular vesicles loaded with the short interference RNA that you can see here labeled in red, and we put in contact these vesicles with the target cells, in particular with colon cancer cells. And what we observe both at confocal microscopy is that this complex is able to be internalized by target cells, and we were also able to quantify, and we obtain a reproducible internalization of these extracellular vesicles loaded with the oligonucleotides into target cells. We then also evaluate what happened to the target. So our aim was to deliver oligonucleotides towards the phospholipase DDHD1 towards colon cancer cells. And what we obtained was that if we treat cells with this complex made of extracellular vesicles loaded with the short interference RNA, we obtain a decrease on the target, so our delivery system was efficient to deliver the short interference RNA into target cells, both at both at RNA but also at protein levels. So what's the next step? We are now studying the protocol for the in vivo validation of this delivery system on mice tumor models. But also thanks to the opportunity of collaborating with the spin-off that, as I said, offer enriched nanovesicles isolated from plants through a patented technology, we want to use these industrial nanovesicles in order to develop our technology also at industrial levels. So this is the team that contribute to the development of this project. So as I said, Riccardo Alessandro, which is the PI of our laboratory, Professor Simona Fontana, as well researcher and professor in our laboratory, and Nima, which is the PhD student mainly involved in this research, together with the spin-off that, as I said, offer us to the project, provide us the vesicles at industrial large-scale production. And thanks to all of you for the attention.
Okay, thank you very much, Stefania. And yeah, we can go to the last speech of the session. Francesco Peri, who is a full professor at the University of Bicocca in the Department of Organic and medicinal chemistry. It's co-founder of a spin-off of the university called CP2 Biotech. So, Francesco, please.
Yeah, thank you very much, Giovanni, for this kind introduction. And yes, as you said, I'm a professor in organic and medicinal chemistry at the University of Milano Bicocca. And sorry, perhaps it's better in the presentation mode. Okay, so this is the title, the name of the patent that was funded by the MISE POC grant, New Synthetic Agonist of Toll-4 Receptor. And what it means, this weird title. This concerns the development of a new vaccine adjuvant. And as you know, as you probably know, the vaccine evolved from the use of the entire pathogen to the use of subunit, so antigens from the pathogen, and also nuclear material. So this increased the safety of the vaccine, because actually you cannot develop infection not being the bacteria or the virus there, but it decreased the immunogenicity. So in several vaccine formulation, especially vaccine, vaccine presenting protein antigens, is needed to add small molecules or polymers that are highly immunogenic, so that are called adjuvant. These molecules help to have a stronger and long-lasting immune response to the antigen. And the role of antigens actually in vaccine is fundamental, and despite this fact, the chemical variety of adjuvant that nowadays are available in the market is pretty limited. And you can imagine that a good adjuvant would improve the potency of the vaccine, so would allow to decrease the dose of antigen that is the expensive part of the vaccine. So this is very important to develop a cheaper vaccine for developing countries, Asia, Africa, and so on. So there is, of course, a need, a global need, I would say, for a vaccine adjuvant. And the ideal optimal adjuvant should have a low production cost and a good activity, a well-defined activity, and a safe toxicity profile. So the story, our story starts here because you know, this guy that is in the middle of the slide is the toll-like receptor 4, and is an essential, a pivotal switch of innate immunity in humans. Is a very important receptor. So if you stimulate this receptor with non-toxic molecules, you get an immunostimulating agent. And on the contrary, if you block this receptor, you can have important therapeutics for inflammatory or even autoimmune syndromes. So in our case, the story started by the MPLA that is represented here in the left. The MPLA is a detoxified version of bacterial lipopolysaccharide that stimulates TOL4. And this is used in important vaccines in the world, such as the vaccine against the hepatitis C and human papilloma virus. So this molecule is pretty complex to synthesize, and actually there is Avanti lipids in the United States that synthesize this compound. But okay, we developed a smaller molecule. Even though you are not a chemist, you can understand that the molecule on the right of the screen is much simpler than the molecule on the left. So it means that we oh, sorry. Did you see still the slide?
We're still seeing the first slide where there is the toll-like receptor 4 receptor.
Okay, because I touched something, I'm no more able to see the slides, sorry.
I see you are presenter. Still, you see presenter. You are presenter. So
yes, I'm presenter, but I'm no more able to see my slide. I don't know.
Francesco, try to stop your presentation and restart it again, so that you can start again from the slides. The system will reset your screen sharing.
Yeah. Okay. But I even.
You do not see the control panel?
Yeah, I can see now the control panel. Yes, sorry.
No worries.
But I'm not more able to go to the slides.
Do you see the screen sharing tab?
Yes.
Okay, stop the presentation and restart it.
Okay. Stop.
Okay.
Okay. And then
Start it again.
I mean, I'm no more in my computer lab screen. Sorry. Okay. Just a moment. Okay. Sorry. Now you can see the slide.
Yes,
it's okay.
Back.
Okay. Sorry. So basically, our molecule is simpler than the MPLA, the monophosphorylipidae, but it has the very similar toxicity and activity profile. And it turns out that the production cost of our molecule is much lower, so it's much cheaper to produce. And so this is basically the advantage of our technology. So before the POC grant, we were in the condition to have these new chemical structures, and this molecule, we characterize very well the mechanism of action, and the industrial, let's say, advantage was that this molecule behave very similarly to MPLA, being much more easy and cheap to synthesize. And the opportunity, as you can see, is the growing interest in the vaccine adjuvant market, especially in the veterinary market that needs very low-cost production for adjuvant. And of course, the drawback was that we had a technology readiness level low, and in particular, we lacked the proof of concept in vivo for the potency and for this molecule to work as vaccine adjuvant. So basically then the POC grant came, and in 21-22 we were able to study the in vivo behavior of our molecule, and we published this paper. So I want to put in evidence that we had also some basic science, and we were able to characterize the mechanism of action of the molecule. Basically, this molecule were published, and several important experiments on animals were done. So the compounds were used in ova vaccination experiment in animal, and as you can see from this graph, after a booster immunization, we got a level of antibody production that was even higher than the level of MPLA. And we also tested the lack of toxicity, so the safety of these molecules. So basically, we started this technology transfer, and thanks to this improvement in the technology level of the molecule, we were able to attract the interest of Avanti Lipid, that is the same company that produces MPLA. And Avanti Lipid was interested to buying the license of our patents. And to manage all this, I founded this biotech, CP2 biotech, that is accredited by Milano Bicocca, by my university. And we are still developing a new a second generation of molecules that are derived from the molecule that I showed you in the previous slide. Okay, so basically, thanks to the licensing of these two patents, we were able to found this company. This is Avanti Lipids, that is the company we are collaborating with. We have a co-development project to have our molecule ready for veterinary market, and then eventually for clinical development. We are developing a very good service agreement with Croda. And basically, sorry, the CP2 therapeutic that was founded by myself, by my colleague Barbara Costa for the biology part. And now we have a CEO that is Sergio Lai, and we have the big wise man that is Fabrizio Conicella, and Giovanni Rizzo knows him very well. And now we have a team, so we were able to hire some PhD students and also postdoc to work on the chemistry and on the biology of the development of this molecule. And basically what I want, sorry, I'm a little bit late, but I had this problem with the computer, so this is the very last slide. What I want to put in evidence is that the spin-off now is based, of course, in the development of agonists, but what we do now is to take advantage from the marketing of, or to the contract with the CRODA on this molecule, also to develop other assets, complementary assets that are the antagonists. So as I told you, the antagonists, the TOL4 antagonists are very important anti-inflammatory drugs that we are developing with a good preclinical track on several autoimmune and also rare disease. So the logic and the business model of the CP2 is to go on with the agonists, but also to develop some basic science and to increase the level of the number of assets. And so going towards the future, always a combination and a good blend between innovation and technology transfer and basic science, good science. Okay, so thank you very much. Sorry for the technical problem that I had, and now I stop the sharing. Thank you very much.
Okay, Francesco, well, thank you very much. So the session stops here at the moment for the speeches. Now there is the panel, and we try to actually to involve also people from the audience. I want to say thank you to the keynote, Italo, and to the speakers, because actually we have been perfect on time. That's something very important for sometimes the challenge to say, you know, you need to stop and everything, but actually we have been quite good. So thank you very much for that. So I would actually open this session for everybody can switch the camera on, and they are actually free to make questions also from the audience. I would start from two questions that actually we got from the audience, and it's about, yeah. Hello?
Giovanni, it was only background noise, no worries.
Okay, I don't have any background noise on myself, but I think there is someone with the open, with the, you know, we should close the microphone, everybody. Okay, perfect. Now we are fine. Okay, so thank you very much. So the two questions that we got from the audience. First, it's not about specifically your projects, but it's about how did you find to make the application for the grant for the POC of the MISE, and what was the requirements and the time that you spend to make the application, and if you would do it again. So that's the first question, and I would make someone to answer. So one of the three that you want to start.
Perhaps the way to go for the different universities are a little bit different. I don't know. I can say for my university, I have to thank a lot of my colleagues of the TTO, because actually this patent was selected. Okay, not all patents were eligible for this grant, because you had to have a certain time frame, okay, in a certain time window, and I had a couple of patents that were included, were eligible, and I found my patents candidates at the level of my university, and then we got the grant. So thank you very much to my TT office, and I want to thank everybody that probably they are also listening to this section. Thank you.
Thank you, Stefania. Alberto, very quickly. Okay, maybe Alberto is already. It's ready. Nobody's ready. Everybody's blocked. Okay. Okay, that's fine. That's the.
Okay.
Now
Okay.
we can.
Yeah.
Now we can.
That doesn't depend on me. Sorry for that.
Yeah.
Yeah. Well, just two words. The process has been very streamlined, I think, not only by our offices, by the respective universities, but also the requirements from MISE were very workable, fair, and so the paperwork was not excessive, and so having the idea, having a clear plan in mind, it was very, convenient and not too much time draining.
Yeah, also in Palermo as well. The application was very easy. Was a two-step application. The documentation was not too heavy, but just a 2-page of application. And actually, we were like informed about it by our research office at the university.
Okay, thank you. So the second question is always on the same field, and actually, it's the question is: do you think more funding opportunities as at the PhD grants could facilitate the research and development projects to move towards entrepreneurship projects spin-off? So clearly, it's a question for all of us. I would start saying that myself as a researcher and as entrepreneurs, when we did the startup, I think actually that's the way to move forward. And I really hope there will be much more grants that, as much as possible, to increase the creation of spin-off from the universities. And on this field, I would actually know one of you the project that applies, and also what Italo thinks, because actually it sees the other way of the ocean. So we're actually, I think the grants for entrepreneurship are really a lot. So who wants to take the questions between the three of us, and then we'll let Italo speak a bit on it.
Actually, I can answer that. Maybe since I am quite close to the academia, but also to the startup environment, I think that there are quite a lot of opportunity in order to do technology transfer and in some way to finance your research in order to go towards a startup. The important thing is to know about it. So I think that a supportive office at the university, but in general in the environment, academic environment, is critical for this, because at European level there are a lot of grant opportunity that I came across with only because of the spin-off, for example.
Perfect. So basically, it's to know.
Giovanni,
yeah.
Sorry, may I introduce some skepticism, because we are very positive and it's correct, but okay, there is money for sure. Can be even more money, because in UK or United States you have normally the research. I mean, the industrial idea starts from university. So this is not so much our attitude to go from academic ideas and innovation to industrial innovation. So it's not so easy in Italy and in Europe. And in my case, by developing new chemical entities, so being in the very challenging pipeline of the drug development, very often what you hear is that, okay, you are too early stage, okay? And so the pre-seed, and also there is another risk. The pre-seed zone is a very dangerous zone, because there is a lot of people that promise you money, but then want this money back, and so it's not easy for a spin-off that is managing this pre-seed level. I would say is a kind of jungle, the pre-seed in Italy.
So let me make this conversation open and discuss openly about this. So the point today, we don't need to discuss if there are the money, how much they are accessible. And I agree with you that actually in the seed, you know that actually there is a new vehicle that has been done by CDP, especially in the seed and pre-seed, which is called Extend. Actually, there are also VCs that are investing ourselves, we invest in seed. So I think actually what Stefania was saying, it's actually interesting. So there are kind of monies, but it's difficult to find it. And there should be perhaps something that let the entrepreneurs to be more aware how to get this money and to unlock that power. And this is one good point. What you were saying is absolutely true. So we need to push a bit more in terms of supporting the let's say the gray zone, which is the seed zone, or the black zone, however we want to call it, but it's fine. But the question was much more on, do you think actually more grants will support entrepreneurs actually to cross that phase? And I think actually it's good, and for sure there is rooms for improvements, that's for sure. But still, I think something is getting better in Italy, and we need to work to make this much even better than what is today. So in terms of Italo, in terms of what Stefania actually was saying, that I feel that is something interesting, you know, how researchers in the US have this kind of access and free access to grants and public grants compared to the Italy, or perhaps if you know the rest of Europe. But what do you think about it?
Well, there's nothing free as a general rule, but the situation is definitely different in the United States. So, and it's changing compared when I started, 20 years ago, 25 years ago. So it's easy to get started. I give you that. The NIH funds small research program in biotech and give you a certain amount of money, not a lot, maybe like 50, 80 to $100,000. That basically covers the expense, the legal fees for the patent, and some of the beginning of the project. But then you need to find money to to advance your research. And then there are different ways. There's definitely out of the academic world, you have tremendous amount of money. So on this I gave you, it's very easy. You have venture capital, you have angel money. If friends and family, if we are in the environment like I am, where technology is pushing the field, it's very easy to find investors to bring up your project. On the other side, what's happening in the last 5 or 10 years, that the university is getting smart. And so big places like Harvard, UCLA, and Johns Hopkins, these are public data, I'm not making this stuff up. If you are faculty and you sign a contract with them as faculty, none of your intellectual property belongs to you, or in reasonable amounts. Most of them becomes to UCLA or UCSF or Harvard, which is incredible. I remember when I signed as faculty at Harvard, I filled out a stack of paper like this big, which is written by Harvard lawyer. There's no way that you make one penny or something you invent. So, and that's why also people are moving out of these places. But so in the academic world, you can find lots of support for your idea. Definitely find, you have the lab, you have the super smart people to do your work, to do the work that you're supposed to do, testing, going from an idea to basic science in animal lab, and then do other type of testing. So you find all that. But the counterpart that's to retain the money of your invention is extremely difficult in big centers, and that's what some of us go through. So kind of a different environment. But outside of the world, venture capital, angel money, and other are very, very readily available in the United States. Absolutely.
So you bring up a very, very sensitive point. What in Italy we call the professor privilege. That actually it's in Europe, it's only in Italy and Sweden, where actually the professor, they have the full ownership of the patent, but the government try to change the rules and making this work. Because, for instance, in the UCL, University College London, you don't get anything, meaning 100% of the intellectual property perspective from the UCL business, but you have the power to negotiate the licensing as inventor, so you can get the patent back and you can make a business out of it, getting the UCL actually will come in the new spin out with equities and supporting you with also funding. So, and well, so there is good and bad, but I think the part that the university needs to lead the way to develop a product is the way to move forward. Otherwise, sometimes, you make the patent and then you stuck there, not doing anything anymore. There is always the other side of the medal. Anyway, I want to continue going straight to the projects now. So that was the main questions from the audience. And continuing with you, Italo, we have questions from the audience, but also I have a couple of questions. There is always a noise that comes along, and I don't know where it comes from, but you can unmute. Okay. So, and the first question is about the aneurysm. So, you were talking about the aneurysm as first clinical, clinical indication. And the thing is that other than an intervention, is there any way that you can reduce at least the risk? And also if there is a genetic behind what happened?
Well, there would be great to have a medication that shrinks aneurysm. We wouldn't have that. So far, the treatment is either open surgery, which is getting less and less done, or endovascular, excluding aneurysm endovascularly. So I'm not aware of any. I mean, there is some ideas of using some type of laser fluorimetry probes to perhaps induce scarring tissue formation, but we're very, very early stage. So far, it's just basically direct contact, either by open surgery or by endovascular. In regards to genetics, usually there are some diseases like collagen vascular diseases that might predispose to aneurysms or family history. So if you have multiple aneurysms in multiple members of the family, there's a genetic predisposition, but most of them are spontaneous, unfortunately. Mostly in women for some reason. But
okay.
I mean, but the great news of the last 20 years, at least in most places, I don't know how it is in Italy. Last time I presented this data in Italy was maybe like 5 years ago. I was attacked by a neurosurgeon that says, "Oh, you'll never replace open surgery," which is completely wrong, obviously. At the University of Rome, it would say in the States it's probably 90% endovascular and 10% open surgery. So we almost not do open surgery anymore, which is amazing. Think about it.
Okay.
It's all technology driven, because you need technology to repair endovascularly, coil stents, catheters, etc. We won't be able to do that.
Okay, thank you very much. And the second question from my side, then I leave also the panel to discuss, and also the question from the audience. It was on acute stroke, actually. You spoke very well, and one thing that made me thinking is that you said, when the acute stroke is coming, then you need to act very fast. So the question is, how fast to avoid the degeneration and mortality of neurons? Because, many times we get a lot of things about post-stroke intervention, and the question that us as investors, we say, well, how much fast should be that intervention to reduce that, to avoid that many risks can happen? And the second question I make straightforward, it's about if any digital tools that at the moment there are many other companies that are coming and say, digitally we can actually see that a stroke is coming or something. So what do you think about that?
So in terms of fast, yes, you have to be fast. The procedure itself, the mechanical thrombectomy, what we do, so entering the artery at the groin, go up and remove the clot, that's pretty fast. I think in high volume surgical center is probably about maybe like 20 minutes or 30 minutes. So it's difficult to shorten that. I mean, we're working on it. I mean, there's the better device, but it's not going to be much shorter. The major delay is from stroke onset to going to the hospital.
The hospital, exactly.
And it's a problem. So in the States, the way we solved it, and I was a president of the Study of Vascular Interventional Neurology, and I work in other company, American Heart, etc. So we devised a system of care based on comprehensive stroke center. So if you have a system that ensures that by the time you get called, the ambulance or the helicopter goes to your hospital, and then the patient gets imaged, scanned, is evaluated, and goes to the angio room for thrombectomy, is within certain limit, then you get granted the stamp of comprehensive level 1 comprehensive stroke center. If you don't get a comprehensive stroke center. Something in Italy is not. So there's an external association called Joint Commission for Healthcare Accreditation that does every 2 years periodically examines all the hospital system and grants this certificate. If you don't have this certificate, then the patient is not. The emergency medical system doesn't bring the patient. But something would be great to have in Italy, but in Italy, unfortunately, it doesn't happen. So when someone has a stroke, it's not that the ambulance says, okay, no, I'll take you to hospital X, Y, and Z because they're the best and they're fast. It doesn't happen because there's no such system. So I envision something like this in Italy because otherwise the delays are terrible. I mean, if you go to a major university medical center, most likely you're going to have good outcomes, but there's no rules on this. And what was the other question?
Yeah. No, sorry, how long it takes basically when you get into the ambulance? I mean, is there anything that you can help to avoid before going to the hospital that this can progress?
Yeah, that's my great question. Yes, ideally, the medication I'm working on and other people working on medication would be beautiful to give it in the ambulance. So the EMS make a prognosis most likely of a stroke. Because now there's the so-called FAST system: face, arm, speech, and time. So you get points. If you have face weakness, and that's very easy to do now. Face, arm is weak, and the speech is slurred, then more likely you have a stroke. So at this point, you hang the medication in the ambulance by the emergency medical system, and then by the time the patient reaches the hospital, the medication is going, and you slow down for expansion. Yeah, that's be great. They tried with magnesium, but magnesium doesn't work. It was a little bit too simplistic. But yeah, ideally, that's a great idea. Once you reach the hospital, there's a very super fast artificial intelligence-based imaging. So that basically, as soon as CT is done and the CTA is done, all of us, members of the team, the neurologist, the interventionalist, critical care specialist, on our cell phone, we got the picture of the brain with the artery block. So everybody instantaneously, you don't have to call the radiologist, or says, oh yeah, there's an MC occlusion. Oh no, there's a penumbra. Oh yes, maybe not, maybe yes. Everybody gets it. You can speak online on the cell phone. And so that speeds up fast, very fast. So artificial intelligence is being used for that.
Okay.
Well, let's think.
Yeah, and then we have actually one question. Basically, it's one question from the audience, which says, how long does it take to bring a procedures from the first try, so the first in man, to the routinary hospital procedures?
I mean, as fast as possible. So Rizzo, we have a hospital that affiliated with us. They are in the Florida Keys, which is about maybe 100 kilometers from here, 80 kilometers from here. But then they go through helicopter. So you manage stroke like a trauma. So there's an helicopter, get in the helicopter.
No,
the hospital,
sorry.
Our hospital has 2 helipads. Oh, can you hear me?
Yeah, no, sorry, Italo. I mean, the question it's much more on the development of the technology more than what is the journey of the patient. I mean, how long it takes for when you start the first try on the patient to get the procedures to be adopted in the clinical setting. So I mean, what, 3, 4, 5 years, 10 years, something like that. I don't know.
So the issue is that you're talking about clinical trials. So once the clinical trials results are announced, that the procedure, for instance, in 2015, when we published the results of the thrombectomy trial, then it becomes standard of care immediately. So hospitals have to do that. If you don't do that, there's no.
When you tried from the beginning, how long it takes? You know, meaning, is there any? What is the time? That's the question. How long is the time to get this technology to arrive to the patients from the beginning?
Oh, from bench to bedside.
Yeah, let's say that.
Oh yeah, that's a long journey. I mean, for this oxygen carrier, I've been working on this for the last 5 years because FDA is very tough. So you need to do, you need to do several. FDA has very strict rules in the States. In order to go to a clinical trial in patient, you need to have results done in several certified basic science labs, different ones. All of them have the same results. And then you write the protocol. FDA approves the protocol. Clinical trials is probably the one that takes the longest because it's extremely expensive. And you're talking about, for efficacy, we're talking about 200 to 400 patient clinical trials. So it's going to take at least another year to 2 years to see if this medication is effective or not for stroke. Now we know it's safe. So 6 years for the idea to say is safe. And now it's going maybe another 2 years, for another year and a half, 2 years to the efficacy clinical trial. But unfortunately, it's a long journey because the rules are very strict here. Yeah.
So Stefania, Francesco, Alberto, any other question for Italo? A quick question we can make as a panel, and then.
I have a curiosity, maybe for Italo. So, the importance of animal models in reaching finally to the clinical and therapeutical applications, because in cancer sometimes there is the tendency to bypass maybe animal models in a way that try to do with PDOs, so with patient-derived organoids, and then the idea to jump from those, maybe have PDX mice, and then but to jump directly to clinic. So I think in certain countries this is viewed positively, in others maybe less. So what is your experience with that in general? Of course, your field is not cancer, so it's a little bit different maybe, but so what is the view, the vision in the States for that? And in your case, do the investors need a mouse model and vivo model in order to invest, or is it possible to anticipate and find funds for clinics, so for therapeutics and solutions even before that?
No, that's a great question, Alberto. I mean, I'm not that familiar with the cancer world, but if you have a model that can be applied, so can you bypass the animal bench testing, of course, it'd be great. For stroke, unfortunately, we don't have that. For stroke, we still need to do the most commonly is the right ischemia model that you enter the artery, you close the MCAs, occlusion and perfusion model. So the FDA still mandates that in order to go from bench to trial. I wish maybe to find a way to do like you guys do. You guys are smarter than us, more advanced, to bypass the animal testing on the full animal and basically choose some cells or some sort and then go to clinical trial. But for now, the FDA is very tough for stroke. You need to do, and for aneurysms too. We did the aneurysm, but the aneurysm model is much easier. It's not there that tough. But for to go from the bench to bedside, you need to do the experiments in animal models in several accredited labs. They are certified by the FDA and by a certain type of accreditation committee to be very reliable and to have similar results. And then you can apply for a clinical trial, design a clinical trial.
Okay, thanks.
Yeah, Francesco. Yeah. Yeah,
I think you're muted. Yeah.
Francesco.
Okay, can you hear me? Okay, because they were switching off. Okay, I have just a curiosity about the material, because I have a lot of colleagues that develop every year new materials. So in your wires or coils or stent that you showed, are you using innovative material as well, and or which is the level of innovation in material that you use?
I'm sorry, what kind of material you said? You mentioned? I didn't hear you. What kind of material?
No, I mean when building up your devices, okay, you use, let's say, classical material, or which is the role of new materials that you can use or in your devices? I mean, because there is an intense research in new materials that have new properties, have flexibility or mechanical properties, and I would like to know if in your case you are also using new materials, or let's say classical one, I don't know.
No, that's a great question. That's a biotech question. And this is divided in our field between big companies, publicly traded companies like Stryker, Medtronic, who have an army of engineers that work on improving the devices, making them more flexible. For instance, we're working now on our surface coating for photoverters, better coils. There's another device called Web that it goes inside the generator like a little basket. So there's something is being developed. Can you hear me? Yeah, something is also developed. There's another world of small startup companies that develop new catheters. For instance, I'm working with a couple of them, developing new catheters, and then eventually get the patent and the intellectual property and the device gets bought by a big company, and the startup company gets assimilated into a big, gets bought out. So there's the two systems. Of course, the big companies have lots of money, lots of engineers, but they tend to be slow. I think the most innovation comes from us. In working in smaller company, that we're more agile, we're more come up with better ideas. The bureaucracy is a little more agile, and then you come up with a product, prototype or something, test, and then the big company buys.
Okay, thank you very much, Italo. Now jumping to Alberto. There are two questions, Alberto, from the audience. One is what are the applications other than the one that you showed, ovarian breast cancer, that PHEGI solution can address? That's the first question. Yeah.
So there are several solutions. Now I presented the ones that are patented, and so where we have the granted patent. So on this granted patent, we are working, and we have now solutions for ovarian cancer, using folate receptor as target, EGFR overexpressing cancer. There are many. We are actively working, have spectacular results, but I cannot show them yet, on neuroblastoma, where this is a pediatric cancer with with very little cures available. And then we also have good results on epithelial sarcoma. So we are focusing on these panels where we think we can have we could have a killer application in some of these.
So basically, it can be considered as a platform for many clinical.
But it is a platform, yes. So if there is a need, to target a specific disease, if the pointer, if the targeting molecule is available, a peptide or an antibody, a nanobody, a single chain antibody, then we can engineer it on the phage and test if the phage platform is specific. So think of it as a super antibody, and besides, because you can think of it as a super antibody with very peculiar characteristic, tumor penetrating, spheroid penetrating, and blood-brain barrier penetrating, then you could also think you could use it for theranostics. And we also develop biosensors, again with the concept that this is a super antibody and can be useful for amplifying the signal.
Okay. There is another question. I would suggest to give a quick answer so we can have more time to. So there is another question which is much more generic, which is about at which point you could have arrived if you actually didn't get the grant, the POC grant. That's from the audience. It's not from me. So.
Yes, way much less way worked. As it's a very little. So the POC grant granted a person to work on it, and allowed us to access more, higher preclinical levels. So the only thing we need to do now is mice.
Yeah. So I have a question more technical and specific, and then I'll ask also the panelists to make questions. So it's about actually the safety and the specificity of your technology. So you showed actually that there is it goes to the tumors, but how do you avoid that this technology is going to normal cells and it's not toxic?
Yes, so the technology is not toxic. So even if it goes to normal cells, the vector per se is completely harmless, unless you shine a light or an ultrasound at it. And light and ultrasound can be focused. So we have a double targeting approach. So the phage reaches the target because the target has a given marker, and the phage is targeted towards the marker. This is the first. Some phages may escape and then bind to other things, but they are harmless. And then you irradiate the region of interest, where you say, for example, you have an unresectable tumor or a chemo-resistant tumor or so, and then the phage is activated, and you do only local damage.
Okay. Any question from the panelists? Is there any other question from the audience? I don't see any other question at the moment. So we go to Stefania. And Stefania, there is actually a nice question from the audience that I would have made as well. Which are the advantages to use plants rather than patient-specific vesicles?
Yeah, actually, I work with both. I mean, I work with plant vesicles, but also with mammalian ones. The advantage, of course, is the possibility to produce at large scale, because with plant, we have the advantage to have a huge amount of plant matrix. And also because of the safety of these plant vesicles that must always be controlled. We also perform in the past a clinical trial using these vesicles in order to ensure about the safety. But for sure, the ones from the nature, from plant, are the ones that we normally introduce with our diet. So they normally are in contact with our body.
Okay, thank you. And then I have a question myself. It's much more a challenge than a question. The challenge is, along those years, oligonucleotides, they failed always in the clinical trials. And I saw that you put already an answer there, meaning that they are unstable. But actually also delivery system has been proven not working amazingly because they don't reach the target or they have off-target effects and everything. So question about first, how do you envisage your go-to clinic and to see efficacy? And the second, what about the competition? Because there are a lot of companies there that they're trying to do exactly the same. And then I have a third question that I make you after these two, which is about cancer vaccine.
Yeah. Actually, what we are doing, and in general the scientific community working on this vesicle is doing, is to take advantage of nature in general, because these extracellular vesicles are not synthesized in the lab, are not like liposomes or other nanostructures, but they are normally produced and released by cells, and they normally deliver among cells biological molecules. So, I mean, we are just trying to use this natural and genuine delivery system to deliver what we want to deliver. But their job in nature, when they have been discovered, is to shuttle molecules among cells. So I think that the advantage compared to synthetic nanoparticles is this. And another advantage, and this is something that we want to work on after this project, but is like the continuation of this project, is that also these vesicles, the membrane of this vesicle can be functionalized in order to specific target some tissue. There are a lot of technologies already developed that allow us a little bit like liposomes to functionalize the membrane of these vesicles.
Perfect. Any question from
to have catch your challenge? I mean,
yeah, more or less. I'm concerned.
Let's see in the future. We are working on that.
I'm still concerned about the efficacy of the oligonucleotides more than the vesicles. That I understand that, but that's a kind of challenge that you are going to face, I think. But what about the panelists? Any question from the panelists for Stefania? No question. Everybody said, oh, Francesco. Yeah. You are on mute. Francesco, you're on mute. We cannot hear you.
Okay, sorry. The first question is, do you treat somehow your vesicles so that you have an homogeneous size, or the size is heterogeneous? And the other question is, there is some advantage also in having on the surface of these vesicles something that is biomimetic or is more compatible with, I don't know, with the human body?
So I didn't catch the second part of the question, but for the first part, actually the heterogeneity or homogeneity of the samples depends on the matrix. For example, I have worked both with vesicles from lemon juice, but also with tangerine one. And what I saw is that, for example, those from lemon are more homogeneous inside. We are also working on improving the technology in order to obtain different fractions of these vesicles with the different size, in order to understand if one are more like advantageous to be used compared to the other one. Okay, sorry, I didn't catch for the audio.
No, the second question is simply about the safety and the biocompatibility. If there is an increase for biocompatibility, given the fact that are of vegetal origin respect to artificial liposome or other vesicles.
Yeah, again, the advantage is that normally when we put these vesicles not engineered and not loaded with nothing in contact with mammalian cells, but also with other organisms, because now there are some collaboration working with also bacteria and other organisms. What we saw is that they are normally internalized by cells without having like a functionalization in the membrane. The functionalization, I think, is needed in order to specific direct these vesicles in a particular tissue.
I also have a question for you, Stefania, very quick.
Go ahead.
So what's the payload of these vesicles? So how many molecules can you stick into them? And second, what's the upper size limit of RNAs you tried to veiculate with this vesicles? I mean, now in RNA therapy world, I mean, we are into this. Perhaps the length of the cargo will be very important for future applications.
Yeah. I start with the second point. Actually, we haven't yet considered this part. I mean, we have worked with very short interference RNA, and now we are working with some double strand because of a project on the agricultural field. So this is really unknown yet. We are working on that. While for your first point, this is very important point, because when I start this project, what I said is that for me in general, the most important things was to be able to quantify the amount of loaded molecules, because sometimes in these studies, not all the researchers are really curious, or I mean are really precise in this stuff. So this is the reason why in our experiment we put a lot of controls using also the supernatant that we obtain after the washing of these vesicles, where we have all the free not loaded RNA. And what we observe is that we are able to have a loaded of the 13%. And when I start, I said, okay, it's not a lot, but then looking at the literature is the mean. I mean, in general, we have a range from the 8% to the 20% of loaded possibility.
Yes, but
Okay.
how many molecules are inside?
Yes.
Yeah. I mean, I haven't quantified the amount of the molecules. I can say that compared to how much we put, we have like 20 picomole inside of the short interference RNA.
Okay, thank you very much for that. Alberto, I think you can take the questions offline. I see that you have many, so you can. Oh, that's a good point. It's nice to get into this conversation. So let's go to Francesco now. That is the last in the queue, and there are a couple of questions. One comes directly from me. Another one is from the audience, which is much more generic. So the first one is, actually to like receptor 4 and activation of inflammation. In addition to inflammation, we say yin and yang, so we don't know what we need to do sometimes. So where is the limit? I mean, when you think you should
you mean developing vaccine adjuvants that are safe, not developing too much inflammation? In this respect, TOL4 is a, let's say, challenging target, because you know very well that you immediately go towards hyper. Point is to have a let's say a stimulation effect and well characterized mechanism of action. In the case of our molecule, we characterize very well the pathways that are stimulated by our compounds, and I would say just changing a little bit the structure of the chemical structure of the compound, we got very different effects. So I think that having a product that can be sold somehow, you need to have a very good characterization of all inflammatory pathways and the safety profile and so on, because of course this is an important question. I mean.
Okay, any other question from the panel? Francesco?
I have a question, a curiosity. You mentioned the market for veterinary so vaccinations. So given the hype maybe in the RNA vaccinations today, and also given the developments, some really nice for example, nanoparticle vaccinations for humans, do you think that the adjuvants in the future will be, will become limited in a way, or to the veterinary market, owing to the new technologies developing for humans? Is that your vision?
This is a very good question in the sense that you mean that the RNA or nucleic acid-based vaccine do not need adjuvant. So this is a limitation of the market, of course. So in 2020, were developed some nano-formulated RNA that were very active without the need to be adjuvanted. And this is, of course, a danger for the adjuvant market, but I think that in any case there are a lot of subunit vaccination and vaccine that are still marketed and very important, as the one that I showed, and that need adjuvant, and especially the veterinary market would need cheap adjuvant, and adjuvant easy to manufacture. So this is still a very important market. I cannot, perhaps Giovanni is much more expert than me, but I cannot foresee if the adjuvant story will end in a total way because this RNA vaccine. I see a variety of vaccine nowadays, so I don't think so, but I mean, difficult to predict, I mean.
Well, yeah, for sure, the adjuvant is really a very, very interesting, way to, to boost the immune system to fight tumors. And now, as Francesco was saying, just to make a little comment on this, there have been two results that came out, one from together with Merck, and the second one from biotech with Genentech, one on melanoma and the other one on pancreatic cancer, where the second one is striking. Nobody saw this kind of results in cancer tumors treatment, because those patients are stable. 50% of the patients, they are stable after 2 years. It's huge. Okay? And actually, it's in the field of cancer vaccines. So using virus to make cancer vaccines, and also cancer vaccines. So it's really interesting field, and for sure modulating the immune system. It's for sure a good point where to start from. Okay, so the second question from the audience for Francesco, and actually it's for everybody. I will make a last comment on this, and it's how to build a good team for a startup, and the person of the CEO, the consultant profile, how could they impact the business of the startup? So that's
okay, thank you. Yeah, I start then Alberto and Stefania. Yeah. So the first recommendation is that as a scientist, as a university professor, don't try to be the CEO of your startup. This is the first point. Okay, so you need something external. You have to have an external vision and a market vision that is not our job. Okay, definitely. So our startup changed a little bit, accelerated once we had the idea to hire a CEO that is coming from the pharmaceutical industry. And the other points for building up the team, I think that I showed that we were able to hire a couple of PhD students that are very motivated, and in this context, I can say that the National Recovery Fund, the PNRR, allows companies to finance PhD and doctorates with very favorable economic conditions. So basically, companies pay the half, and they can develop doctorate, industrial doctorate into the university. So this was another positive point, in the sense that I was very pleased to have the opportunity to hire motivated PhD student by this law, let's say this, this financial support from the PNRR. And finally, I think that as in every team, you have to differentiate. I mean, in our case, you have to keep different person for doing what is the industrial development and the basic research. So both of these aspects you need to have, but again, I think that the expertise is slightly different. So don't try to be to have the same person for doing basic research and industrial development.
Stefania, thank you, Francesco.
Yeah, I completely agree with Francesco. I think that one of the major points is that one academic cannot do everything, or if he try, he cannot. So an important thing, since a lot of spin-off start from the academia, is to have a balance between the academic figures and the others. In our case, for example, it was very important to have an industrial partner, because the industrial partner give us the opportunity to have possible commercial view, so contact, and also another way to think to the things. And then another critical point, I think, is to identify from the start the consultant, because it's impossible to have all the figures necessary for the development of a startup inside, but it's important to identify from the first moment all the other figures, all the other skills that is very important. For example, legal advice, intellectual property advice, all these I think that should be with a startup from the beginning to all the steps.
Perfect. Thank you, Stefania. Alberto.
Yes, well, I'm really fresh in my experience, so I can say if our startup, our spin-off will be successful or not. But for us, it was really important to understand that we will go the same way. So we have the same expectations out, you know, for the spin-off, so the same vision, and then to invest and not to use you know, PhD students or postdocs that are working on our academic projects on this, but to invest and make clear that this is completely, you know, it's not academia, and we need to hire persons and stuff that is definitely 100% on the spin-off, and then our lab will just back up with the technology and the knowledge. But in a way, the spin-off should try from scratch, you know, hiring new people that are dedicated to it, and therefore grow up with a spin-off idea and not from academia.
Okay, perfect. Well, I think there are no other questions. I just want to answer to this, just to wrap up and to make a comment on this. I've been on both sides, and basically I agree that the researcher, they should look at people that they can read in the books, and they can understand what is the science, but also what is the business. So, and that's absolutely important. And what is also important is that there should be an advisory board, and there should be people on the board that can help you to drive the, you know, the boat. So meaning investors are not only, I look on the side of the investors, as you know, and as I said, I'm a very technical guy, and investors, they're not people just financing you, but they're people that are trying to give you the tools for you to move on, not only financial, but also in terms of networking and also expertise and experience. So at the end of the story, you should have a board of directors which is can help you to drive the boat, and at the same time a scientific advisory board that can support you in the decision-making process, especially on what you are going to target as you can imagine, clinical indication more than preclinical indication and everything, and how this transition can come. So yeah, the wrap up. So I would actually go into the wrap up basically, and I want to say thank you very much, first of all, to NetBulk for organizing this amazing sharing event. Actually, it's very important. I would suggest anybody to continue to follow the tech co-leader, and there is the next session that actually is really interesting, which is innovation in Italian healthcare, and they're speaking about challenges and opportunities. So whoever is on the call can join the other session just with a different link that has been provided to you. And thank you very much to the speakers, to the organizing, to inviting us, and to have this nice moment. So good luck to the entrepreneurs, and thank you very much, everybody.
Thank you.
Bye. Thank you.
Bye.
Bye.
Thank you.